I hope you have a good break. Now let's get back into the urinary system. So we know the site of the kidneys and size of the kidneys, and we know the function of the kidneys. So one of the functions of the kidney is excretion of the waste products from our blood. Their job is to filter and excrete. That is only one of the many functions of the kidney. The other function will be like endocrine function and electrolyte imbalance function. So if only for the one function, which is the filtration and secretion, if we want to convert into a machine, these are the machines we are looking at. Yeah. So these machines, these are the renal dialysis machine, which is the equivalent for the kidneys in our body. And if you think about that, you know, how efficient our kidneys are. Look at the size of the kidney, what they can do, what these machines can do, and what our kidneys can do. Is our kidneys are still far better. I just want you to appreciate it. And of course, what are the structures making our kidneys to this useful? Of course, the nephron, nephron. So this is the new terminology which I want you to pick up. Nephrons are the workhorse for the kidneys, and kidney without nephrons doesn't mean anything. And therefore, nephrons are important. These are the functional units of the kidney. So if you remember the cortical radiate artery, from the cortical radiate artery, a small little artery, which we call arteriole, which is coming into the socket, which we call it glomerular capsule. So this afferent arteriole coming into the glomerular capsule, where they become capillaries for the filtration, and leave the glomerular capsule as the efferent arterioles. So look at the size. Afferent arteriole is larger and efferent arteriole is smaller. And here we have our glomerular capillaries, which is inside the glomerular capsule. And that is one portion of the kidney, which consists of the glomerular capillaries and glomerular capsule, or in other words, it is also called Bowman's capsule. And combination of this capillary and capsule, it is called renal corpuscle. So renal corpuscle plus the tubular portion, so tubular portion of the kidney, it forms the nephron. So at the level of the glomerular capillaries and glomerular capsule, there will be filtration, and anything filtered, there will be absorption into the peritubular capillaries, or there will be secretion from the peritubular capillaries. That is the basic mechanics, basic function of the nephron to form urine. So I want you to remember all these things. The neurons are the functional unit of nervous system. And the nephrons are the functional unit of the kidney. Sarcomere, they are the functional unit of the skeletal muscle and or structural unit of the skeletal muscle, and the osteons are the structural unit of the compact bones. And this is the nephron, which include the glomerular capillary and glomerular capsule, which makes the renal corpuscle and the tubular portion. And where do we get these capillaries? It is from the afferent arteriole coming from the cortical radiate artery and leaving the capillaries efferent arteriole, and they will continue and they will loop around the renal tubules, and therefore we will call them peritubular capillaries. And finally, they will go back into the cortico-radiate vein. Nephron is the functional and structural unit of the kidneys. It includes renal corpuscle and renal tubules. Renal corpuscle, as you have seen, it includes glomerular capillaries and glomerular capsule, which is called Bowman's capsule. In the tubular portion, we have the loop, which we call loop of Henle, collecting ducts, proximal convoluted tubule, and distal convoluted tubule. So that makes a nephron, which includes renal corpuscle and renal tubule. The filtration rate in the nephrons is like 1 liter per every minute. It is quite a good filtration. And nephrons are mainly located in the renal cortex. So the glomerular capsule plus the proximal convoluted tubule, distal convoluted tubules, they are all located in the renal cortex. So now I want to explain you how a nephron formed. So this is the renal corpuscle, which is made of the capillaries and the Bowman's capsule. And after the renal corpuscle, we got our proximal convoluted tubule, then loop of Henle, then distal convoluted tubule, which we drain into the collecting tubules. So the way it forms is the glomerular capillary is being invaginated into the Bowman's capsule. If you look at this Bowman's capsule and this if you push the glomerular capillaries push into the Bowman's capsule, now here we get this. Yeah. So by doing so, we have the parietal layer of the Bowman's capsule, which is made of the squamous epithelium. And the visceral layer, which is made of cells, they are called podocytes. So glomerular capsule parietal layer is made of the flat squamous epithelium, and the glomerular capsule visceral layer is made of the podocytes. Yeah, podocytes, these are the resting on the basement membrane. So if you remember the 3 types of capillaries, they are continuous, fenestrated, and the sinusoidal capillaries. In the kidneys, renal glomerular capillaries are made of fenestrated capillaries. So now we have created a Bowman's capsule. The parietal layer is made of the squamous epithelium, as you can see, and our afferent arteriole coming into the Bowman's capsule became the glomerular capillaries, and then finally leave the glomerular capillary as efferent arteriole, and the visceral layer of the Bowman's capsule is made of the podocytes. Here you can see the podocyte cells looping around the fenestrated capillaries. So I hope you understand podocytes, food processes, filtration slits, and filtration membrane. So now let's talk about the filtration membrane. So here on the capillary side, we have the plasma, and we have the capillary endothelium, which is again like a flat squamous epithelium, which is to sit on the basement membrane, which is colored with green. And from the Bowman's capsule side, we have the podocytes with its food processes. And here, the plasma is filtered across this membrane, and these are the filtration slits and the These are the food processes of the podocytes. That is the way our blood filtered into the Bowman's capsule. And now this is the photo micrograph, which is showing the podocyte, which is surrounding the glomerular capillaries. So as we understand, all the arteries come in and the veins come out, efferent arterioles become glomerular capillaries, become efferent arterioles, and then they will continue around the renal tubules as peritubular capillaries and vasa recta, which is a strict vessels, and finally leave as cortical radiate artery. Of course, we have cortical radiate artery. Here we have the cortical radiate vein. So now I said most of the nephron is in the cortex. So if you look at this here, this is the outer portion, which we call cortex, and this is the inner portion of the kidney, which we call medulla. And this here, this is the corticomedullary junction. And this is the cortical portion, this is the medullary portion. You can see the arcuate artery here. From the arcuate artery, we have the cortical radiant artery coming out. From the cortical radiant artery, efferent arteriole come in, go into the renal Bowman's capsule as glomerular capillaries, leave the glomerular capillaries as the efferent arteriole. Yeah, and now we study glomerular capsule or Bowman's capsule plus the glomerular capillaries. That is what we call, yes, it is the renal corpuscle, and from renal corpuscle, proximal convoluted tubule, which is in the cortex portion of the kidney, and then medulla go into the medulla as loop. And here you can see thin loop and slightly bigger, thick loop, and come back into the cortical portion as distal convoluted tubule, and finally comes into the collecting ducts here. And therefore, the renal corpuscle, proximal convoluted tubule, distal convoluted tubules, they are in the cortical portion of the kidney, and loop of Henle, it is in the medullary portion of the kidney. And of course, renal tubules, because they have the secretory function and absorptive function, and therefore they are made of cuboidal epithelium. But nephron loop, thin loop, is made of the flat squamous epithelium. And the collecting ducts, again, they are made of cuboidal epithelium. So now this is more of a complex diagram. We know that here, this is the arcuate artery, which is running in the corticomedullary junction. From the corticomedullary arcuate artery, it comes the cortical radiant artery. From the cortical radiant artery, we have our efferent arteriole, which goes into the Bowman's capsule. And then from the Bowman's capsule, then the glomerular capillary leave and become the efferent arteriole. And efferent arteriole will continue as peritubular capillaries. And some of the peritubular capillaries, they might not be looping around the proximal and distal convoluted tubule. They will be running with the loop of Henle as vasa recta, meaning straight vessel. So there are two capillaries. And here again, you can see we have two types of nephrons, which are cortical nephron and just the medullary nephron. What does it mean? Cortical nephron, most of the portion of the nephron is in the cortical portion. Only the small portion of the loop is in the medullary portion. And juxtamedullary nephron. Juxtamedullary nephrons have long loop of Henle, which are remaining in most of its parts in the medulla. Of course, whatever it is, the proximal and distal convoluted tubules remain in the cortical portion. And 85% of the cortical nephrons, 85% makes the cortical nephron, and only the 15% makes the juxtamedullary nephron. Now look at that. This is the vasa recta, which is running along the loop of Henle, and capillaries in the kidney. 99% of the glomerula filtrate. So these capillary will filtrate into the Bowman's capsule, but 99% of the filtrate will be reabsorbed. As we know, there are two types of capillaries which are mix and match around the proximal and distal convoluted tubule. They are called peritubular capillaries, and those capillaries which are running along with the loop of Henle, they are called vasa recta. And the peritubular capillaries plays a role in low pressure system and for absorption and secretion. And vasa recta will play a role in concentrating urine. Of course, now we can see the afferent arterioles, efferent arterioles, glomerular capillaries. So now let's keep looking at more detail around the afferent arteriole, and the Bowman's capsule here. This is the afferent arteriole coming into the Bowman's capsule and finally for the glomerular capillaries and leaving the glomerular capillary as the efferent arteriole. And now you can see the capillaries, which are fenestrated capillaries, which are cycle around by the podocytes here, as we have seen before. And this, at any cross section here, this is the ascending limb of the loop of nephron, and might be the distal convoluted tubule here. Here we have a cell which is close to the afferent and efferent arteriole on the wall of the loop of nephron. We call that cell macula densa cell here. And if you look carefully here, afferent arteriole coming into the Bowman's capsule, and now the cells here got thickened here. That is what we call granular cells. And now in between afferent and efferent arteriole, together with the loop here, we have formed a little triangular space area where there are a few little cells which we call extraglomerular mesangial cells. So now we have seen the macula densa cell, extraglomerular mesangial cell, and the granular cell of the afferent arterioles. Why am I talking about these? These are called juxtaglomerular cell. Juxta means close to, glomerular means glomerulus. So close, the cells close to the glomerulus, they are called juxtaglomerular cells. They are macula densa, juxtaglomerular cells, and extraglomerular mesangial cells. So I want you to concentrate here. The macula densa, they are the cells of distal convoluted tubule or the loop of Henle. They act as a chemoreceptor and they monitor the solute concentration in the filtrate, and as appropriate, it stimulates granular cell to secrete renin. So this macula densa here, macula densa, they do not secrete renin, they detect the concentration of electrolytes in the tubule, and depending on the concentration, they will ask the granular cell to produce renin. So granular cells will produce renin. It is under the influence of macula densa. It also contains the mechanoreceptors. Extraglomerular mesangial cells, they interact with the macula densa and G cell. So now I hope we can understand this diagram where the afferent arteriole, the cells become thickened here as it coming into the Bowman's capsule. These are called granular cells, and leaving efferent glomerula. And now we have the ascending limb and distal convoluted tubule. Here we have the macula densa cell, which act as a chemoreceptor. And now we have the extraglomerular mesangial cell, and the granular cells will secrete renin. Let's take a break and we'll go to the next chapter.